video, I'm going to give you an overview of the plant cell and do so under the context of how plant cell structure creates the necessity of the differences between plant and animal lifestyles. So, in common with animal cells, plants have a plasma membrane, mitochondria, smooth and rough endoplasmic reticulum, Golgi, cytoskeleton, nucleus, etc. But plant cells may also possess plasmodesmata, connections between these cells, unique to plants. Cell walls, the structures that create plant cell shape and structural strength. Chloroplasts, probably what you think of when you think of the major difference between plant and animal cells is the chloroplast, and structures called vacuoles. So chloroplasts are just one of many plant-specific structures called plastids. Another is the amyloplast, a starch-storing organelle. Chloroplasts are really crucial, though. Hey, they are what make plants green. But more importantly, they're what allow plants to be organisms that do not consume calories, even though they burn calories. So plants convert light energy into chemical energy. That's where they get their calories. Plants do not consume calories. The stuff that they get from the soil is not used as a calorie source. Plants have cell walls. It's sad to me that they're called cell walls. I believe they should be called cell fences. So, a primary cell wall consists of cellulose microfibrils and hemicellulose crosslinks between these cell fibrils, a little bit of glue thrown up in the form of pectin. So it's outside of the plasma membrane, this primary cell wall. It's visible by microscopy as a set of microfibrils. It looks almost like woven cloth, I think. And those fibrils are this material called cellulose, which is a series of glucose molecules in a linear form that hydrogen bonds with neighboring cellulose molecules. And these hydrogen bonded together molecules form the microfibrils. The microfibrils are several molecules, all hydrogen bonded together, providing enormous tensile strength. It's remarkable that sugar can be the substance that provides the hardness of wood, isn't it? So, if you were to think conceptually about what these cellulose microfibrils are doing, it helps to know a little bit about construction techniques. So, rebar, that's the pieces of iron, steel, that you find laid down before workers pour concrete. Now, a primary cell wall doesn't have a concrete analog, but it does have these microcellulose fibrils that are analogous to rebar, because that's where the tensile strength comes from. That's what keeps concrete from falling apart. Concrete is great at resisting compressive force, but terrible at resisting pulling apart force. That's what the rebar is for. That's what the cellulose is for. A primary cell wall then has the cellulose microfibrils, has the pectin, has the hemicellulose crosslinks that tie the cellulose together, but lacks this other material, which is called lignin. There's a couple of pictures of primary cell walls. Most of the cells in a plant are parenchyma, and parenchyma simply means cells with only primary cell walls. plant cells with secondary cell walls are referred to as sclerenchyma. So, primary cell walls into which the plant cell injects lignin become secondary cell walls. They're not really an additional structure, they're an addition to the structure. Now, an interesting thing about cells with secondary walls, or sclerenchyma, is that they are waterproof. I said that cell walls ought to be called fences. That makes sense for primary cell walls, like a structure made out of just rebar. It's a three-dimensional fence. There's plenty of room for even large macromolecules to travel and for water to travel freely. But if you add lignin, if you now have secondary walls, you have a waterproof structure. Remember that no cell can survive if it's not surrounded by water. If your cell is waterproof, it's going to die. And the strange thing about sclerenchyma cells is their function really begins after they've died. It's the laying down of the secondary wall structure, and the secondary wall structure itself, that provides the function that these cells add to the plant's repertoire of mechanisms. So the vacuole is a space within the cytoplasm that is not the cytoplasm. It's a space whose contents do not have to be maintained with normal homeostatic conditions. The enzymes that are functioning in the cytoplasm don't have to function in the space within the vacuolar space. That provides this huge volume within the cell that does not have to be maintained under comfortable conditions. You could, and very often do, have a very acidic vacuole, for instance, so that the plant's enzymes are not affected when you suddenly import a tremendous amount of water. Now, if you were to do that into the cytoplasm, that would dilute the contents of the cytoplasm. The proteins wouldn't function correctly. You instead have a different place, another bag into which you can throw this water, and now you can import water magnificent rate, which allows really fast plant cell growth. Plant cells will import water into their vacuole, and that vacuole will expand and expand and stretch the cell out, which consists of growth. For putting pressure on the walls of the cell to generate expansion of a cell requires that the cell be capable of expansion, requires that it be plastic. Now, some cells are elastic. Elastic means you can stretch them out and they'll return to their original shape. Plastic means you can stretch them out and they'll stay at the new shape. Growing cells, expanding cells, are plastic. The reason they're plastic lies in the orientation of the cellulose microfibrils of the cells. If they're all lined up in the same direction, forming rings around the cell, I've drawn them as rings in this upper right diagram, is cutting the quick links between the microfibrils, which are arranged as belts, all going in the same orientation around these cells, and these fibers, which in this drawing are close together, in this drawing are far apart, are maintaining the size of the cell in one dimension, not restraining the size of the cell in another, so that the cell can grow in a particular direction, expand in a particular direction. And that direction has previously been dictated by how those microfibrils are laid down. The direction of cell division, too, is determined by the laying down of cellulose microfibrils. When plant cells divide, they don't pinch off, as do many animal cells. Instead, what happens is the nucleus divides, and then a wall is built between the two daughter cells. When a plant cell divides into a pair of daughter cells, this shared cell wall, owned by both cells, binds these cells together permanently. The wall precludes any movement away from one another, which is a pretty significant restriction in terms of mechanisms of development. So in animal cells, you'll see many occasions in which cells start off next to each other and end up having new neighbors a little later in development, and that proximity to new cells changes their function, changes their fate, but not so in plants. Once a cell has a set of neighbors, it has those neighbors for life. Let's look at this cell wall that connects a pair of cells. Cell walls in plants have structures called plasmodesmata that allow movement of materials through from one cell into the other. Now, this connection between adjacent cells is so profound that some plant biologists argue that there is no such thing as separate plant cells. They're just a continuous structure with some interference in the middle of them, alike to, say, having a tennis racket in the middle of a fountain of water. So, I'll edit that out. Some plant biologists even go so far as to make the claim that plants don't have individual separable cells. Why? Well, the plant cell membrane is here indicated by the blue line, the cell wall by the orange painted line or structure there. So this is a two-dimensional drawing of a three-dimensional structure. These look from a 90-degree angle, to be a circle. If I were to turn this 90 degrees so it faced you, through this hole between the membranes, you'll find endoplasmic reticulum sticking through. And this endoplasmic reticulum is part of the endomembrane system. And remember, the endomembrane system consists of the endoplasmic reticulum and also the nuclear membrane. All of this forms each time the new cells are formed. And they're continuous. This membrane system is continuous between the two cells. The membrane is continuous between the two cells. The cytoplasm, one could even argue, is continuous between these cells. So if you were to try to move one cell with respect to another, you'd be ripping the endoplasmic reticulum, you'd be ripping the cell membrane, and the cytoplasm would be severed as well. Consequently, plant cells do not move with respect to their neighbors. What sorts of questions might I ask you about this video? Well, I can't think of any off the top of my head.